I hope you guys would be fine. And now here, this is the second lecture for the biological molecules. And earlier I have said that the biological molecule, they have the atom and these atoms, they would be grouped with one another to be making the molecules. And among the biological molecule, now we look on toward the proteins. As for this word protein is to be considered, so this is a polymer of the small monomers that we have with us. So these are the amino acids. So if I take the amino acids, if I combine them with one another, if I condense them together, so as a result of that, we would make protein out of it. Now we must know that what elements we need to have in order to be making these proteins. So we need carbon, we need hydrogen, we need oxygen, and in addition to these three elements that I have written for you, so we would be in the need of the fourth element, and this fourth element that I have with me, so this is nitrogen. So in addition to this, nitrogen is something differently which is coming into the protein. Now the scenario is that where in our body we have got these proteins or what could be the functions of these proteins that they would be rendering in our body. If you see, we have got the red blood cells and the red blood cells they bind with oxygen gas or they carry the oxygen gas around in the body. So in the red blood cells we have this protein hemoglobin. Likewise, if I have to make the nails or the skins or the hairs of my body, so for that I require another protein and that is keratin with me. And at some point you may have got ill and the doctors may have said that you have got some infection of the pathogens. So if in my body if I have the pathogens and if I need to be removing them off or I need to be eliminating them off, so I need this protein and this is antibody protein. And earlier, we know that in our human body, there happens the metabolism of the glucose where we convert the excess glucose to the glycogen or the glycogen back into the glucose. So for that, we require these protein hormones and we have the insulin and the glucagon. We are alive for a reason that multiple chemical reactions would be catalyzed within the cells or within our bodies, and for that purpose, we require the enzymes. So these are again the proteins with us. And along with that, these proteins that we have, they would not make all of the cell membrane, but the major part of our cell membrane, they would be made up of these proteins now. Now we must know that how general formation of the protein is going to take place. So I need to take the monomers. These monomers are the amino acids. These amino acids, when they would be combined or linked with one another or condensed with one another, like I have to be linking them by the elimination of water, so we would end up making the polypeptide chains. And these polypeptide chains, further, they would make the proteins out of it. So this is how the general formation of the protein would take place. Like I need to take this amino acid, then amino acid, linking them to be making the polypeptide chains, and we would end up making proteins out of it. I have forgotten some of the other, you could say, role of the protein that whenever the repairing of the damaged part of the bodies has to be done. So these are the proteins. And we also make the tendons and the ligaments with these proteins as well. Now moving onward, the situation is that how does the amino acid look like? So our body total of possesses 20 amino acids. And in those 20 amino acids, we would have this carbon, we will have this hydrogen, we will have this carboxylic group. Does the carboxylic group looks like? That is C double bond O, and then I have to be linking it to this OH group. So this would make the carboxylic acid group. Then we would have this hydrogen, and along with that, if I were to add this NH2 group with this carbon, so this would be the amine group. So this carboxylic group, this hydrogen atom, this carbon atom, and this amine group, so this is common in all of the 20 amino acid. Each of the 20 amino acid, they would have a different group in them, and that different group is termed as or entitled as the variable group. And this makes every of the amino acid different from one another. So you must note it down that all the amino acids that we have in our body, they would have a different variable or the R group. Now, at any cost, at this level, you must not know how you're required to be drawing them up, but you must be very much aware that what groups we have got in the structure of an amino acid. Now, the scenario is that how I am required to be linking the amino acids together. So I would make a separate diagram for you. So you see, I would take this carbon. To this carbon, I am linking this carboxylic acid group. then I would have this hydrogen atom, then I would have this amine group with me, and then I would have this variable R group, and we are leading to the formation of a protein. So now there comes another amino acid, and here again I have got this group. This group is the amine group, and then I need to link it to the carbon. Here comes a hydrogen. At the top we have the variable group, and on this side we have got again this carboxylic acid group. So from the carboxylic acid group, there would be removal of OH group. And for the amine group that we have here, so there would be elimination, or there would be the removal of the hydrogen atom. And they both would be making water out of it. Now, since I have to be removing them off, so let me just simply erase them. So when I am erasing them, so the situation becomes like this. Okay. And now we are gonna be making this peptide bond here. So this carbon bond, I am elongating it, and then I am connecting it to this nitrogen atom, and then here comes this hydrogen atom. So this linkage of C double bond O and NH is what we refer to this as a peptide bond. And when multiple proteins, they would be linked with one another, multiple of the amino acid would be linked with one another with the help of the peptide bond. So we would end up making the polypeptide. So this is the same diagram. I have one amino acid. This is the second amino acid. From the carboxylic acid group, I have to be removing this OH group. From the amine group, I am required to be removing this hydrogen atom, and then we would end up making this peptide bond here, and this peptide bond goes as C double bond O NNH. And here we have made up the polypeptide chain, and we have eliminated the water molecule out of here. And when these multiple polypeptide chains, when they would be linked with one another, so we would end up making proteins out of it. Now, the opposite happens, like if I have the protein and if I have to be reverting them back into the amino acid, so I need to add water into proteins. When the water would be added up into the protein, so we would make the polypeptide chains. And these polypeptide chains, then they would be reverted back into the amino acids. So I hope your elaboration of the protein would be clear now.